get right to it. So I'm going to spend the next few minutes talking about a novel immunotherapeutic target that we discovered in the laboratory through multiple orthogonal approaches that we think holds potential to enhance immune responsiveness in prostate cancer. Here are my disclosures. So we initially did an unbiased screen using single-cell RNA-seq data from metastatic prostate cancer samples with bone metastases. And what we discovered, strikingly, was that the NLRP3 inflammasome was highly expressed within the macrophages and the monocytic lineages within the bone metastatic prostate cancer patients. And these are patients that were treated with ADT. They were both MS castrate sensitive as well as castrate resistant patients. For those of you that are not thinking about the inflammasome, it's a multimolecular complex that's typically turned on by a priming mechanism that involves LPS or pattern-associated molecular patterns. So this is, could be, secondary to infection, inflammation that then drives priming of the NLRP3 monomer through NF-kappa B. And then there is a second step, which is an activation step that results in oligomerization of the inflammasome. And that then results in inflammatory cytokine release in a phenomenon called pyroptosis through gasdermin D that releases these molecules and essentially lights the fire for inflammation. So what we discovered, actually, interestingly, was that these inflammatory monocytes and macrophages from the metastatic patients, we were typically we saw this upregulation in the prostate cancer patients that had bone metastases, not the untreated primary prostate cancers. And when we looked across multiple tumor types, we didn't see any significant increase in the inflammasome. So that led to the hypothesis that perhaps androgen deprivation therapy was in some way driving the upregulation of this molecule in human patient samples. So we took a reverse translational approach, came back to the lab, and then essentially took in vitro macrophages that we force differentiated into an M1 versus M2 lineage. And we were struck by the striking difference between NLRP3 expression, which is very high in the anti-tumor M1 macrophages and low in the M2 macrophages. And conversely, we saw high levels of AR in the M2 macrophages and low in the M1 macrophages. That then led to the hypothesis that AR blockade enhances NLRP3 expression within these M2 tumor-promoting macrophages in the microenvironment. So we went back again to a bioinformatic approach, and in collaboration with Amy Miran's group at Oregon Health Science, we were able to access the Stand Up to Cancer West Coast Dream Team data, as well as data from a clinical trial of MCRPC in patients that had progressed on enzalutamide. And what we discovered was that there was a really interesting negative correlation between AR activity and NLRP3 inflammasome activity across multiple metastatic sites in these enzalutamide or abiraterone-treated MCRPC patients. We then went on to do single-cell RNA-seq on samples from Medicure in collaboration with Alok Tiwari and others at the Dana-Farber, and essentially looked at signatures of M1 versus M2. And what we found was that the M1 times correlate with high NLRP3 expression, and that doesn't really change before or after ADT. On the other hand, the M2 times actually see a significant increase in NLRP3 expression following ADT. We also then reverse translationally came back in vitro, looked at our immortalized bone marrow-derived macrophages and found that ADT or AR blockade in these macrophages actually drives NLRP3 expression in the M2 TAMs. And this was then corroborated by QRT-PCR data looking at M2 versus M1 TAMs, where we see the upregulation of the target with AR blockade, specifically in the tumor-promoting macrophages, not in the anti-tumor M1s and not in the MCAP tumor cells. So this was unique to the M2 TAMs. We then collaborated with Michael Hafner here at the Hutch to look at some ChIP-seq data and essentially demonstrated very interestingly that the treatment with androgens actually induced binding of AR at H3K27 acetylated mark sites across the NLRP3 gene locus in a human macrophage THP1 line. And we then went on to show that this upregulation of NLRP3 is actually functionally relevant, and we can actually activate the inflammasome pathway when we give an NLRP3 agonist, which we obtained in collaboration with Bristol Myers Squibb that was developing the NLRP3 agonist at that point, and we were able to leverage our International Immuno-Oncology Network collaboration to look at that. So the next question is, does this really matter? And, if we're upregulating this complex in the M2s, does that functionally change, how these macrophages behave? And so our hypothesis is that this could potentially serve as a macrophage phagocytic checkpoint. And through a series of experiments that I don't have time to go into right now, we discovered strikingly that, there was not only an upregulation of the molecule in these M2s, but these macrophages now, in combination with NLRP3 agonists, potently induce phagocytosis and upregulate CD86 and PDL1. The M1 macrophages, on the other hand, can directly be activated with the NLRP3 agonists alone without ADT. But the sum total of this, when you give this in combination, is you potently drive phagocytosis and tumor control, which was what we wanted to understand next. And so we went back to our in vivo murine model, which is a c-Myc-driven model, to ask the question of whether this activation actually promotes or reverses immunosuppression and eradicates murine prostate cancer. So this is the punchline on the in vivo data, where we essentially treated our MICAP tumors with the combination. And as shown here, 60% of the mice actually showed complete tumor control. And when we gave clodronate, which is a macrophage depleter, we significantly attenuated the anti-tumor response, suggesting that this effect was driven through activation of macrophages, initially driven by priming through ADT and then activation. So this is, now, an area of major focus in the lab is developing these, we're collaborating with Argonne National Lab to develop novel systemic activators that we're going to then hopefully move into the clinic at some point. And this is just concluding that NLRP3 inflammasome is an AR-regulated macrophage checkpoint that can be inducibly expressed within these TAMs following AR blockade, resulting in phagocytosis. We think these therapeutic strategies hold promise, mostly in combination with IO strategies. In the context of a PCF Challenge Award, we're actually looking at these questions, taking a deeper dive into the epigenetic mechanism and also looking at combinations. So I'll stop and take questions. Thanks.